Hello, my name is Dr. Venon Sofa, and in today's video, we're going to talk about the Cori cycle. So this is going to be a short video. But if you enjoy this video, make sure to subscribe to my YouTube channel so that every time I post a video, you are going to get notified. So without wasting your time, let us get started. So first of all, we need to ask ourselves, what is the Cori cycle? So the Cori cycle is simply a biochemical pathway that integrates anaerobic respiration that is taking place in the muscles with gluconeogenesis taking place in the liver cells. So if you look at this diagram, you're going to see that on this side we have got the muscle, and then here we have got the liver. So what is happening in the muscle? So in the muscles, we are using glucose. So this glucose, where is it coming from? Glucose, it can come from diet. It can also come from glycogen breakdown. So in the muscles, when we break down this glucose through glycolysis, this glucose is going to end up producing what we call pyruvate. Now, when talking about pyruvate, that pyruvate, it has got two pathways where it can go. Because pyruvate, it can enter into what we call Krebs cycle. And that can only happen if there is the oxygen. Now, in the absence of oxygen, you're going to find that pyruvate is going to be transformed into what we call lactate. Now, this lactate, you need to understand that it cannot be utilized in the muscles. So it must be taken to the liver. So it's going to be transported through blood all the way to the liver. And then in the liver there, lactate is going to be converted back to pyruvate. When it's converted to pyruvate, this pyruvate is going to be utilized in Krebs cycle in the liver, but most of this pyruvate that is coming from lactate is going to be transformed back into glucose. And this process of converting lactate into glucose is what we call gluconeogenesis. And then this process of producing lactate in the muscles is what we call anaerobic respiration. So in the muscles, we have got anaerobic respiration, and then in the liver, we have got gluconeogenesis. So in the liver, we are going to produce glucose, and this glucose produced in the liver is going to be released into blood. And then in blood, it's going to be taken back to the muscles, where it's going to be utilized again in glycolysis. So this cycle, it's what we are calling the Cori cycle. So when we talk about the Cori cycle, we are simply trying to connect anaerobic respiration taking place in the muscles with gluconeogenesis taking place in the liver cells. So let's look at this in more details. So if you look at here, you can see that on this side, we have got glycolysis taking place in the muscles. And then here we have got gluconeogenesis taking place in the liver cells. So in the muscles, we know we are going to have glucose. This glucose, like I said earlier on, it can come from glycogenolysis, the breakdown of glycogen. Because glycogen breakdown in the muscles, it ends up to glucose 6-phosphate. In the muscles, we don't have gluconeogenesis. The reason is simple. Because the muscles cannot convert glucose 6-phosphate to glucose. Why? The muscles do not have an enzyme called glucose 6-phosphatase. This enzyme is only present in the liver cells. You're going to find that glycogenolysis, it's going to end up to glucose 6-phosphate. When you make glucose 6-phosphate in the muscles, this glucose 6-phosphate is going to enter glycolysis. When it enters glycolysis, we're going to end up producing pyruvate. This pyruvate, it can be utilized in the Krebs cycle or TCS cycle. But if you look at the muscles are always contracting and they are always in need of ATP. So you're going to find that sometimes oxygen is not going to be sufficient for us to be able to convert pyruvate into acetyl-CoA. So you're going to find that in the absence of enough oxygen, some of the pyruvate is going to be converted to what we call lactate. Now, this lactate, when it's produced, lactate is actually lactic acid. So lactic acid, when it's produced, you need to know that it can cause acidosis. And this can lead to muscle cramps. But this lactic acid is going to enter blood. When it enters blood, it's going to be transported to the liver cells. So when it's transported to the liver cells, what is happening? So in the liver here, this lactate is going to be converted back to pyruvate through an enzyme that we call lactate dehydrogenase. So it's going to be converted to pyruvate, and then pyruvate is going to be converted back to glucose in what we call gluconeogenesis. This glucose, when it is produced in the liver, it's going to be released in blood, and then it's going to come back to the muscles where it can be reutilized in glycolysis. So basically, this is what we're talking about. This is the Cori cycle. Okay, now let us just quickly look at glycolysis. So glycolysis, as we can see, we have got glucose being converted to glucose 6-phosphate. So let's imagine this is in the muscles by an enzyme called exokinase or exokinase. And then glucose 6-phosphate is converted to fructose 6-phosphate by an enzyme called phosphoglucose isomerase. And then fructose 6-phosphate is converted to fructose 1,6 bisphosphate by an enzyme called phosphofructokinase 1. Then this molecule is converted to glyceraldehyde 3-phosphate and also dihydroxyacetone 3-phosphate. Dihydroxyacetone 3-phosphate cannot be utilized in the glycolytic pathway. So it must be converted to glyceraldehyde 3-phosphate by an enzyme called triose phosphate isomerase. And then glyceraldehyde 3-phosphate is going to be converted to what we call 1,3 bisphosphoglycerate. This is done by an enzyme that we call glyceraldehyde 3-phosphate hydrogenase. And then from there, we are going to find that this molecule is going to be converted to 3-phosphoglycerate by an enzyme called phosphoglycerate kinase. And then from there, we are going to convert this molecule to 2-phosphoglycerate by an enzyme called mutase. And then this molecule is going to be converted to phosphoenolpyruvate by enolase, and phosphoenolpyruvate to be converted to pyruvate by pyruvate kinase. So depending whether we have got oxygen or not, or don't have oxygen in the muscles, if we have got oxygen in the muscles, this pyruvate will be further converted to acetyl-CoA by an enzyme called pyruvate dehydrogenase. But if we don't have oxygen, this pyruvate is going to end up being converted to what we call lactic acid. So converted to lactic acid by an enzyme called the lactate dehydrogenase. This lactic acid that is produced in the muscles, if it builds up in the muscles, it can cause muscle cramps. So it's going to cause denaturation of the myosin and the actin filaments, which can result in muscle cramps. And this lactic acid, it can be transported through blood all the way to the liver. And then in the liver there, what's going to happen? We are going to have the reverse of glycolysis. The reverse of glycolysis, we call it gluconeogenesis. So if you look at this diagram, you can see that here on this side we've got glycolysis and here we've got gluconeogenesis. So what's happening in gluconeogenesis? So you can see that in gluconeogenesis, we are going to start with what we call pyruvate. So we can start with pyruvate. This pyruvate is going to be converted to what we call oxaloacetate by an enzyme called pyruvate carboxylase. And then oxaloacetate is going to be converted to what we call phosphoenolpyruvate by an enzyme called phosphoenolpyruvate carboxykinase. The reason we are trying to move around at this step, it's simple. It's because this step is an irreversible step of glycolysis. Okay? So if you remember, phosphoenolpyruvate to pyruvate in glycolysis, it's an irreversible step. So we can't move direct from pyruvate to phosphoenolpyruvate. We must first move from pyruvate to oxaloacetate and then to phosphoenolpyruvate. These reactions, okay, these are reversible reactions. So we're just going to use the same enzymes. So phosphoenolpyruvate is going to become 2-phosphoglycerate by the same enzyme enolase. 2-phosphoglycerate is going to become 3-phosphoglycerate by the same enzyme the mutase, and so on and so forth. So the same thing is going to happen. So the only difference that you're going to see between glycolysis and gluconeogenesis is at the steps that are not reversible. The irreversible steps, you're going to see difference. All the reversible steps, everything remains the same. So at the end, we are going to produce what we call glucose 6-phosphate. Now this glucose 6-phosphate, it must be converted to glucose by an enzyme called glucose 6-phosphatase. This enzyme, it is not there in the muscle cells. It's only found in the liver. So it's found in the endoplasmic reticulum of the liver cells. So this enzyme is going to convert glucose 6-phosphate to glucose. And when we make glucose in the liver, this glucose is going to be released into circulation. When it's released in circulation, where is it going to? It's going to go to the muscles, because the muscles, they are using a lot of glucose each and every time. So it's going to go back to the muscles, and in the muscles there, it's going to be utilized again in glycolysis. It will be broken down, and we can end up again producing lactic acid, which can come back again to the liver, and the cycle continues. So this is what we are calling the Cori cycle. If you enjoyed this video, please make sure that you subscribe. Thank you so much for watching.